Hi, this is Dr. Malescu. This is just a quick on the assignment that is a bonus. It's worth 10 points. And basically, it has to do with the topic of cell structure and function that we are doing this week, week 2 in lab, and week 3 in lecture. So you can find this in the assignments folder. And some of you have already started the process of answering these questions, so I'm going to delineate the guidelines and let you know that it's not just one or two sentences. It is a process, it is an essay in explaining to your best knowledge with critical thinking skills, using your own words, not copying and pasting and sourcing off the internet. I would like for you to learn the process with your own language concept asked of you in this uh, assignment. Okay, so the first question: explain the cell cycle to include details of the cell cycle stages, including the mitotic phase. So what we are talking about here is basically explaining the cell cycle, which starts out as G1, S, and G2. So you could look up what happens in G1, what happens in S phase, which is basically DNA synthesis, and G2. So you have to elaborate what happens in those steps. And then mitotic phase, you describe the main highlights of what happens in each phase. So in phase, what happens is the nuclear envelope disappears and starts to develop. Towards the end, late prophase, you're going to start to see the spindle forming, so that chromosome preparing to move to the center. The next phase to the center, the cell, that's metaphase. Anaphase, to the opposite polar ends of the cell. So you start to develop two. Telophase is the phase where the nuclear envelope reforms, the nucleolus becomes visible again, the chromosomes become chromatin, and then cytokinesis occurs where there's cell division. So that's question number 1. Question number 2, you have to elaborate between active and passive transport. So passive transport goes with the concentration gradient from high to low. It's like rolling the ball down the hill. There's no effort. Whereas active transport is pushing the ball, requires energy. So passive transport does not require ATP, which is the form of energy that the cell needs, which is made by the mitochondria. And then active transport is going against the gradient where ATP is needed. Because we're going from concentration to a high concentration, whereas passive goes from a high concentration to a lower concentration of that molecule, that ion, or whatever we're talking about. Okay? Now, in addition, you also have to elaborate the 3 different types of passive and then the different types of active. So types of passive transport is simple diffusion, facilitated diffusion, and osmosis. You need to elaborate the difference between. You have to go over the active transport, where you have sodium potassium pump, 3-to-1 ratio, where sodium diffuses in, potassium leaves. All right, at a 3-to-2 ratio. And then you have the exocytosis and endocytosis. These are the 3 types. You need them. All right, so look that up and really figure out how you're going to word it. We will be discussing it in both virtual classroom for the lecture. Hopefully that will give you the answer. What is apoptosis? Apoptosis is cell suicide. Cell suicide literally ends and the cell commits, no longer continues. If it continues to divide unabated and considers itself a cell that continues to divide and divide and divide. All right, so that's answered. Now, number 4 is protein synthesis. You need to explain the details of transcript, the details of translation. The detail is that the DNA travels slightly at a area of a helix, double helix, and the mRNA reads the naked DNA strand. It transcribes it, it reads the genetic code. The code is a stop codon, so that is released from the DNA and then leaves the nucleoplasm via the nuclear pore, enters and attaches to the rough endoplasmic reticulum. Now it's part 2, translation, where the mRNA 2 ribosomal units translation occurs. During this time, the ribosome moves 3 codons at a time, which is the genetic code that has been transcribed. RNA has this codon. It's a genetic message, 3 nucleotide bases. And so the tRNA that's going to come in is going to have a complementary sequence that matches with those on the mRNA. The anticodon binds with the codon, complementary sequencing of nucleotides, and releases its amino acid. It goes away, and another tRNA comes in as the ribosome, another set of 3 nucleotides, another codon is translated, and another tRNA comes in. Its complementary sequence binds to the codon, and another amino acid will bind to the previous amino acid, and so on and so forth. tRNA comes in and releases an amino acid. You build up a chain of amino acids called a polypeptide chain. At the end of the mRNA, there's a stop codon again, and the polypeptide chain of amino acids is released. And it goes back into the nucleoplasm to undergo another endless cycle of making protein. The ribosome stays where it's at because it's bound to the membrane, the rough ER, and that newly formed peptide chain will travel over to the Golgi apparatus where packaging and delivery. The package or the polypeptide gets folded up quaternary into a legitimate protein. It then gets transported out via the Golgi vesicle. It attaches to the cell membrane, invaginates, and spits it out via exocytosis. ATP is used, and now the protein is out of the cell. Number 4. Number 5, what are the steps to DNA synthesis? You can look there's there's a short story. I want the short story bullet format. Basically, it's the DNA unwrapping and replicating itself, a pair pairs of the same genetic material. So eventually, the next cell, the 2 equal daughter cells, they will be the same genetic material as before. In humans, we have 46 chromosomes, so the DNA unravels. You got 1 strand here, 1 strand here, and so on the right here, pretend that the nucleotides are forming, so you got a double helix here, and then the second DNA strand that unraveled, another buildup of DNA here